staining and bending the chromosomes. Chromosome staining procedures has been developed that help to study the different regions in the chromosome or the bending patterns of the chromosome. These techniques have helped to study the chromosomes which are present in the plant cells as well as in the animal cells. Here we can see a chromosome at a metaphase where in the middle there is a centromere and there are dark bands and there are light bands. So this is called as chromosome bending pattern. By using different dyes, the regions on the chromosome can be stained. Some of them appear as dark bands and some of them appear as light bands. Bending types. There are 4 most common bending types: G bending type, C bending type, R bending type, and Q bending type. Here is the example of C bending type. C bending type is for bending the centromere. We can see in the diagrams that the centromere has been stained and it appears in the form of a dark band. This type of bending where centromeres have been stained, the type of the bending is called as C bending. Human chromosomes can also be stained and they also be studied for the karyotyping. In case of humans, there are 46 chromosomes that consist of 23 pairs. Out of these 23 pairs, 22 they are called as autosomes and 1 pair of chromosomes is called as sex chromosomes. Here is the bending pattern of human chromosomes. We can see that all the homologous chromosomes which are present in a diploid cell, they have been arranged. This is the pair of chromosome 1, then chromosome 2 and so on. Here is the chromosome X and here is the chromosome Y. From this bending pattern, we can say that this bending pattern belongs to the male chromosome bending pattern. This diagram shows the human chromosome bending pattern where all the chromosomes they are showing light bands as well as dark bands. As there are 4 different types of the bending pattern, the first bending pattern is Q bending, which is performed with the help of a chemical. The name of that chemical is quinacrine. In case of Q bending, usually the chromosome arms they show the bending pattern and which shows that there is a AT rich region. In case of G bending, which is performed with the help of a dye, the name of the dye is Giemsa. In case of Giemsa staining or in case of G bending, chromosome arms they are stained. While in case of R bending, there is a variety of techniques which is used to perform the R bending. In this case, again, chromosome arms they show the bending pattern. But in case of R bending, that is specific for GC rich region of the chromosomes. The 4th type of the bending pattern is C bending. C bending, to perform the C bending, there are different techniques and C bending is particularly for the centromere of the chromosomes. So we can say that there are different bending patterns of the chromosomes. 4 Important bending patterns, they are G bending, C bending, Q bending as well as R bending.